It's 12:30. I think I'll start because there's a lot I would like to do today. First of all, we're going to be talking about cell structure and function. And in the textbook, the relevant pages for that are 55 to 76. And I'll put that, when I send you the Zoom link to this session, I'll include those page numbers so you'll have them handy. I have assigned another worksheet on today's topic, cell structure and function, and it's due on Wednesday at noon, just before class, so that you will have done it by Wednesday and we can discuss it in class. In addition, there will be another quiz, quiz 2. This one will be for real. I'll grade it. And it will be due on this coming Saturday, the 12th. But you'll have any time between now, today, I think I made it open today, anytime between now and noon on the 12th to do quiz 2. And that will cover everything we've covered so far. Introduction to biology, methods of scientific inquiry, and cell structure and function, today's topic. So that quiz hasn't been assigned yet. I haven't composed it yet, but I will. So you'll have it available well before Saturday. So the first thing I'd like to do today is discuss quiz 1. Do you see quiz 1 on your screen?
Yeah.
Okay. This wasn't graded, and the point of this was to familiarize you with the types of quizzes that I give and give you an opportunity to check out your equipment, your electronic equipment. So let's just go over the questions and the answers quickly. First one, true or false? Biology is one of the natural sciences. It is a branch of science that deals with physical phenomena on planet Earth. That is false because it doesn't deal with physical phenomena. It deals with biological, living phenomena. The second, which of the following is not a property of life? Well, I've highlighted here in yellow the answers: heat absorption, combustibility, homeopathy, and transmissibility are not properties of life. All the others are. Then 3 is an example of a matching question. Energy processing is matched with locomotion. Single-celled is the simplest form of life. A toad is a highly organized set of cells, tissues, and organs. A polar bear is an example of adaptation to extreme cold. And a California redwood tree is a case of extreme growth. And then there are 3 additional possible answers that you had to make sure you didn't choose. Number 4, the smallest fundamental unit of matter is the atom. And then I asked you to write a short statement. This is an example of an essay question. An example of how knowledge of biology impacts your daily life. And what would be the result if that knowledge were not available? So this, and I say less than 100 words. In all essays, I will usually limit the number of words because I will always be asking for short answers. And this was up to you to think about how biology impacts your daily life, how you deal with it every day. The next one is multiple answers. Indicate which features are common to both theory and hypothesis. Well, they are falsifiable and testable. All of the others here are not the case. And then 7 is another true or false. A controlled experiment is one in which everything is held constant except for one variable, and that's true. And finally, on the graph, indicate the axis representing the dependent variable and the independent variable. And the answer to that is shown here. Time on the x-axis would be the independent variable. And on the y-axis, the velocity would be the dependent variable. Any questions about the quiz, the example quiz? Either the answers or the format, or did anybody have problems accessing it or performing it?
No problems here.
Okay.
I just have a question regarding the essay questions. You said for this one in 100 words or less. In the future for quizzes and exams, will you let us know about how many words you want?
Yes. I will almost always indicate that what the word limit would be. And if I do, then I won't read beyond that number of words.
Okay, thank you.
All right, then I'm going to stop sharing this screen. Now you should see structure of life. Yes?
Yes.
Okay. Cell structure and function. We're going to talk about prokaryotes, eukaryotes, and viruses. What is a cell? Well, the smallest unit of life. And I put a big circle around cell theory, as I'd like you to remember this. It will surely come up again. A cell theory has 3 components. First, all living organisms are composed of cells, 1 or more. The cell is the basic unit of life. And importantly, cells always arise from pre-existing cells, in contrast to the theory of spontaneous generations. So this is important for you to remember. Also important is that viruses are not cells. They're sometimes described as acellular, but they fall in a completely different category. They're not considered to be alive. And we'll talk about why in a little bit. Now, I'd like to deal with the scale of things, scale of size, and consider how do we see very small things. So what you see here from left to right is very small to very large. So if we start on the left, an atom is very small, usually less than 1/10 of a nanometer. Molecules are groups of atoms put together, so they're larger. And lipids, for example, are large molecules. Proteins are even larger. And here's a virus, a collection of molecules. And then we have bigger things like organelles, mitochondria, chloroplasts, which are smaller than bacteria, which are smaller than plant and animal cells. And then we get to see the size of cells that we can see with our unaided eyes. For example, frogs' eggs, chicken eggs, ostrich eggs, hummingbirds, humans, and finally, a giant redwood. So the things to the right here, we can see without assistance. To the left, we can't see. Until there were microscopes, we didn't know these things existed. And so we're going to talk a little bit about the types of microscopes you need to see these very small things. Now, there are a couple more points that I want to make here. One is, you'll notice that the scale here is in micrometers or nanometers or meters. This is the metric system. It's used in science around the world, and it's used in most countries. However, not in the US. In the US, we use what's called the imperial system, where a meter, instead of being 100 centimeters, it's 39 inches, which is the equivalent of 3.3 feet, which is the equivalent of 1.1 yards. And we actually use all of these measurements in our daily lives. For example, in football, the markers are in yards, not in meters. And this is unfortunate because there are only 3 countries in the entire world that use the imperial system. The other 2 are Liberia and Myanmar. The US actually tried the metric system for a short period from 1975 to 1981, but then it was abandoned and we went back to the imperial system because the public really didn't like it. They didn't like going to a gas station and buying their gas in liters. So in the US, at least for the foreseeable future, we're stuck with the imperial system in everyday life, even though we use the metric system in science. And this is required. If we couldn't use the metric system in science, it would be virtually impossible to do science. So I just wanted to make that distinction and let you know that the US is aware of the metric system and has chosen not to use it, except for scientists. And now I'd like to show you a short video which gives you a further discussion of size. Everybody see this slide with pictures
Yeah.
of cells? Okay.
Yes.
All right.
natural to just categorize a whole series of things as just being really, really unimaginably small. So when people say, hey, atomic scale or molecular scale or protein or a cell, you often just group that together and say, oh, those are really, 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 really small things. But what I want to do in this video is get an appreciation that even though all the things I just mentioned are really small, there's actually a huge difference in the sizes of those things. And hopefully that'll give us an appreciation for how complex something like a cell can be, how it can have all of this machinery, how it can actually be a living organism or part of a living organism. And so at this scale, this is my little rendering, my drawing of a water molecule. You have the oxygen atom in the right over here in this purplish color, and then you have 2 hydrogen molecules bonded to it. And this is going to be roughly 0.275 nanometers. And just to remind ourselves, a nanometer is a billionth of a meter. And just to get an appreciation of that, so this is 1 billionth, 1 billionth. 1 billionth of a 1 billionth of a meter. That's a nanometer. And if you want to even attempt to visualize that, that would be a millionth of a millimeter. So let me write this, 1 millionth of a millimeter. And I actually like using this one because a millimeter is about as small as I can on a reasonable basis visualize, but it's in a millionth of that. So this definitely goes well beyond at least my capabilities of visualization. So that would be the diameter or the width of a water molecule. But now let's go to the next scale up. We've talked a lot about proteins, and this is our friend, this right over here is our friend hemoglobin. And just so you get a sense of scale, the width of hemoglobin is going to be about 5 nanometers, or 5 billionths of a meter. Now that seems super small, so in some ways it's okay to categorize that into your super small part of the brain, but it's good to appreciate this is much larger than a water molecule. If a water molecule were on this scale, I pre-drew it, this little thing over here, that's my attempt at drawing a water molecule at this same scale. So even when you go from something like a water molecule to a protein, you're already going dramatically up in size and dramatically up in complexity. And we've talked a lot about protein structure, how they can take on all these interesting shapes and do fairly surprising and complex things inside of biological systems. But now let's go the next scale up. And the next scale up, I'm going to go to a virus. And what I've attempted to draw here, this is a fairly well-known virus. This is HIV, and it's actually one of the larger viruses, and its diameter is roughly 120 nanometers. So if we were to draw this hemoglobin protein at the same scale as we've drawn this virus, this thing right over here would be the hemoglobin protein, and we wouldn't even be able to see the water molecule at this scale right over here. But this is still really, really small. This is 120 billionths of a meter. So this is still still unimaginably small. But now let's go up to the next scale. So this creepy picture right over here, this is a T cell. This is a depiction. This is if you want to see the whole thing, that is a T cell right over here. This is a T cell. And this creepy picture, all the blue, that's the T cell. And what you see in yellow, that's the HIV virus emerging, taking advantage of this T cell. So that's why it's so creepy. It's using that cell's machinery to reproduce itself. But you immediately see on this picture how small the HIV virus is compared to the actual T cell. Each of these small things is an HIV virus, which we already saw is a lot bigger than something like a hemoglobin protein. And so hemoglobin protein, you wouldn't even be able to, on this scale, maybe it would be a pixel, if that. And on a similar scale as this T cell, you have things like red blood cells. And this is actually a comparison, side by side. This is using an electron microscope. You see a red blood cell right over here, and you see a T cell. And they're roughly on the same size, or at least the same order of magnitude size. And a red blood cell is going to be 6 to 8 micrometers wide. So this is 6 to 8 millionths of a meter. So if we were to if we were to just take 7 as the average, 7 millionths, 7 millionths of a meter. Over here, we're talking about a millionth of a millimeter. Now we're talking about 7 millionths of a meter. And just to get an appreciation for size, we already compared the virus, the HIV virus, to this cell. We're seeing it directly as they emerge from the cell. But each of these red blood cells are going to contain roughly 280 million hemoglobin molecules. So there's going to be 200, each of these, there's going to be 280 million of these. So 280 million, that's not million hemoglobins in each one of these. So hopefully this starts to give you an appreciation for, even though we categorize cells as these unimaginably small things, they're actually far larger. They're ginormous compared to things even like large, even proteins, and especially when you think of things on the molecular or the atomic scale. And that's why cells are so interesting. They actually have a lot of complexity to them. But just to have an appreciation also for how small cells are, even though we've just described these red blood cells and these T cells, there's these kind of worlds unto themselves. They're these incredibly complex things. If I were to draw the width of a human hair on this screen right now, relative to the scale of these red blood cells, it would be about as wide as this video. So if I were to draw a human hair, it would go from there roughly to there. There's actually a lot of variance in the width of a human hair. But the width of a human hair would be just about like that if you looked at the scale of this picture right over here. If you looked at these scales, it would be much bigger. And I encourage you, we can think, oh, okay, width of a human hair. Pluck a hair out, look at it, put it on a piece of paper. It's hard to even discern the width. But we're saying that width compared to these red blood cells would actually be the entire width of the screen. We've already said these red blood cells, these C cells, these are worlds unto themselves when you think of it from a viral or especially at a molecular scale.
Are you back to the slides?
Yep.
You can see it? Okay. Yeah, so I hope that gives you a little deeper appreciation for the difference in sizes of things. And you notice that he mentioned microscopes. So now we're going to talk a little bit about the sizes of things and how you see them with microscopes. So one type of microscope is called a light microscope. A compound light microscope is fairly sophisticated. And for our purposes, what you do with this type of microscope is look inside of cells at a fairly gross level. So what you see here are 4 plant cells. And what a light microscope will show are the large things inside a cell. So the big dark object is a nucleus. The smaller green things are chloroplasts. The big open area is a vacuole. And you can see the cell wall. And that's about it. So you get a good look at a plant cell with a light microscope. But if instead you use an electron microscope, which operates on a completely different principle, a transmission electron microscope shows you the inside of cells, the fine structure of the interior of any object. So here you have the same 4 cells, so 4 plant cells. And you can see the nucleus and the chloroplast and a lot of other things as well, which we'll talk about later. The point I want to leave you with is that a transmission electron microscope shows you the fine structure inside of objects. Now, in contrast to that, we have a scanning electron microscope, which shows the outside of objects. Doesn't have to be a cell, but for our purposes, we're most interested in cells. So we see the fine structure of the outside of the cell. And you'll notice here, compared to seeing everything on the inside and very little on the outside, with a transmission scope, with a scanning scope, we see nothing on the inside, but great detail on the outside of these same 4 plant cells. So, any question about that? What we use different types of microscopes to see the things that with only our eyes, we can't see at all. So we can see the outside of cells. We can see inside cells at a rather small level. Fine structure inside cells with the transmission scope, and the fine structure of the outside of cells.
I have a question.
Yeah.
What's the difference between a microscope and a telescope? So, I mean, they sort of do the same thing. A telescope, it gets closer to the moon. If you're trying to look at the moon, it gets closer.
Yeah, and that's a good question. It leads to where we're going to go next with this. when we're not looking at very small things and we want to look at very large things that we can't see with our unaided eye, then what do we use? And you are right, we use a telescope. So we'll get to that. This is a cat's tongue. You've all touched a cat's tongue. You know it feels like sandpaper. And this is why, because all the cells on the surface of a cat's tongue come to a point. And this is taken with a scanning electron microscope, which shows you surfaces. Now, back to Esteban's question. If we go beyond our planet to look at things we can't see with our unaided eye, we can use a telescope in the backyard. Here's the Palomar 200-inch scope. Here's a radio telescope. And now we have space telescopes, which escape from the problems caused by Earth's atmosphere. They go out into space and take pictures. And we'll talk more about what space telescopes can show later, but just for an example, here's a picture of the Milky Way, our galaxy. In this yellow spot is the center. Our sun, in other words, us, we're way out here on the edge, a very small part of the galaxy. This galaxy contains about 300 billion stars. And if we assume that every star has at least one planet, then there's roughly 300 billion planets. Probably more than that. But it doesn't matter, this is such a huge number. But the thing I want to emphasize now is distance in the universe, because this is going to be a limiting factor when we start talking about life elsewhere. Okay, so for example, the Milky Way has a diameter of 200,000 light years. What is a light year? It's the distance that light travels in a year. How fast does light travel? 186,000 miles a second. So if you multiply that by the number of seconds in a year, you come up with nearly 6 trillion miles across our galaxy. And this is just one galaxy in the universe. If you look at this picture on the right, which is an image taken from a space telescope of just one very small slice of the sky, it contains 15,000 galaxies. So imagine how big our galaxy is. And all of these other galaxies, 15,000 of them of various different sizes, many of them much larger than our galaxy. So it boggles our minds to try to think of what the universe is really like, because of the numbers of things that are there and the distance that these objects are apart from each other. And I have another slide set, which goes on from here, life in the universe. I don't know if I'll get to that today. I'll try, but before doing that, I want to make sure I finish talking about cell structure and function. So back to biology. What kinds of cells do we see in nature? Of two types, prokaryotic and eukaryotic. The prokaryotes are bacteria and archaea. Eukaryotes are plants and animals. And then we have another category, viruses, which are not cells. So prokaryotic cells are the simplest. They're very small, usually a single cell. Their genome is DNA. They have no nucleus, but rather the DNA is found in what's called a nucleoid region of the cell. And they have no membrane-bound organelles. They do have one type of organelle, the ribosome, but it's not bound in a membrane like in eukaryotic cells. So this is prokaryotes. And the bacteria are usually found in one of three different shapes. Either a rod, called a bacillus, a sphere, called a coccus, or a spiral shape, called a spirillum. Now, what do we see if we look inside a bacterial cell? On the upper left is an E. coli bacterium cell. And you see the DNA is this long chain of nucleotides. It has a cytoplasm, it has a cell membrane, actually has a cell wall, which is labeled here. Usually they have flagella. And I'd like to mention, as in passing, an amazing thing about DNA. And that is that this cell, bacterial cell, is about 0.003 millimeters long. It's pretty small. The DNA inside that cell is about 1 millimeter in length. So the cell is 0.003 millimeters. The DNA is about 300 times longer than the cell itself. So how does it do that? We'll talk about it later, but essentially what it does is twists itself into tiny little highly condensed strands so that it easily fits in a much smaller object than its length itself. In the lower left, you see a cutaway with labels of the things inside a bacterial cell. The nucleoid DNA is a major component. The little red dots are ribosomes that make proteins. And then you have a cell wall here. There's a cell membrane called a plasma membrane, and then flagella. And on the right, a picture, a real picture of bacterial cells and then a drawing of that. So what kind of a microscope, based on what we just talked about, what kind of a microscope took this picture? There are three types of microscope: light microscope, transmission electron microscope, and scanning electron microscope.
Transmission electron microscope?
Yes, you're right. It is a transmission, and the reason we know that is that it's showing the fine structure inside the cell. It's not giving as much detail of the outside of the cell. So this is a transmission electron micrograph. Now we'll switch from prokaryotes to eukaryotes. Eukaryot, eu means true, karyon means nut or nucleus. So eukaryotes have a true nucleus. And remember, I've been emphasizing that prokaryotes don't have a nucleus. They have a nucleoid region where the DNA is kind of floats in the cytoplasm. There are two general types of eukaryotic cells, plants and animals. The genome is always DNA, size from 10 to 100 micrometers. And they've evolved from progenitor cells. And here you see a picture of animal cells, a picture of plant cells. Plant cells have very regular shapes, and that's because they have cell walls, which are rigid. Animal cells don't have cell walls, so they take on various different shapes. In fact, animal cells, these are all animal cells going from muscle to skin to nerve cells to blood. And you can see that they're highly variable in terms of their shape. Well, here's something for you to think about. I'm not going to tell you the answer now, but just help you think about the question. The human body contains about 230 different types of cell. So if you think of your own body, you've got skin cells, you've got blood cells, nerve cells, hair cells, and so on. Now, each of those cells in your body contains about 23,000 genes. And it's the same 23,000 in every cell. So it doesn't matter whether, doesn't matter what kind of a cell. These are cells that are highly variable in both their shape and their function, but each of these has the same 23,000 genes. So can you come up with a hypothesis to explain why these very different cells have the same genetic material? So we'll talk about this later after you've had some time to think about it. So another question is, why are we not made of just one big cell? Why do we have 23,000 cells? Excuse me. Yeah, why do we have 230 different types of cell instead of just one big cell? Humans are made up of trillions of small cells in order to maintain a high surface area to volume ratio. So just to illustrate that, you take this big boxy thing, one cell, surface area of one large cube is 5,400 micrometers. If you take that same volume and divide it into 27 smaller cubes, you now have increased the surface area to 16,200 micrometers. So this results in a much greater efficiency and capability. It would be impossible to have organisms the size of us, for example, if we were just one cell. So by having trillions of small cells, it facilitates transport and communication, cell specialization. So if we have just one big cell, it'd be one type. Instead, as we've just said, we have 230 types. And that's made possible because we have many small cells. And injury repair. If we had one big cell, you injured that cell, the whole organism would be dead. Whereas because we have so many cells, if we injure some of them, they can be discarded and replaced with new cells. You don't destroy the whole organism. Any questions about that?
No questions.
Okay, then we'll go on and talk about the structure of an animal cell. See a drawing of a cell here and a transmission electron micrograph on the right. So we'll talk about most of these different organelles as we go on through the semester, talk about what they do. But one thing I want to contrast is an animal cell from a plant cell. First of all, there is no cell wall. There's a plasma membrane, but no cell wall. There's a lysosome, a centriole, and a flagellum, and plant cells don't have these. What plant cells do have is all the same things that animal cells have, plus a cell wall, plus chloroplasts, and plus a central vacuole. And all of these things are critical for life on Earth because the cell wall is necessary for rigidity so that plants can hold their shape. The central vacuole is necessary because it holds water. And why is that important? Because animals don't have this water bag, because if they're thirsty, they can go find some place to get a drink. Plants can't do that. So when water is available, they store it up in this large vacuole, like a camel stores water in its hump. And finally, plants have chloroplasts. And we'll spend a lot of time later in the semester talking about chloroplasts and what they do, but in essence, they're the only thing that sustains life on this planet because they're the only things that can take the energy in sunlight and convert it into chemical energy. And we'll go into that in great detail later in the semester. And now viruses. They're not cells because they can't reproduce on their own. But we need to understand them because they affect us in many different ways. In fact, the reason we're online instead of in class is because of a virus. Viruses can infect almost any kind of cell, prokaryote and eukaryote. They're very small, and characteristically, they can't reproduce on their own. They have to get inside a cell and use the cell's machinery to reproduce themselves. So here are examples of viruses. Tobacco mosaic virus. These are transmission electron micrographs. Tobacco mosaic virus is a long, rigid rod. This is T4 bacteriophage. So here's the virus, and here's a bacterial cell. The virus is about to invade it. And this is HIV, a spherical virus. And here it is, HIV inside the cell. And what I want to point out in particular, not only are different viruses different in shape, they have different types of genome. So for example, tobacco mosaic virus has not DNA, RNA as its genome. T4 has DNA, just like plants and animals, but HIV has RNA as its genome. So in contrast to real cells, which always have DNA, viruses can have either DNA or RNA. And this makes a profound difference in how they replicate once they get inside the cell. And we'll talk about that later when we go into detail about coronavirus. Speaking of, here are some pictures of coronavirus. The important things I want you to remember about it are it's spherical. And on the surface is what's called a spike glycoprotein. And this is what it needs to infect human cells. And when you hear about variants of the virus that have different levels of infectivity, it's because of mutations in this spike protein. And we'll talk more about that later. And again, I want to emphasize coronavirus is an RNA virus. Its entire genome is made of RNA, and this is critical in the development of vaccines. And we'll talk about vaccines against coronavirus a little later. So let's look at some example problems. You're looking at a cell under a compound light microscope. It's approximately 50 micrometers in diameter. You can see the nucleus, you can see a cell wall. What type of cell is it?
Plant cell?
Okay, plant cell. That could be true. Look at the size. Approximately 50 micrometers. So it could conceivably be a very small plant cell, but in fact, that's very likely what it is, a plant cell. And that's because the nucleus is visible and it has a cell wall. So let's look at another example. This says you're looking at a cell. So we know it's a cell. It's rod-shaped and smaller. This is about 4 micrometers in length. Doesn't have any internal structures, but it does have a cell wall. What type of microscope is being used?
A virus.
The type of microscope.
The transmission electron microscope.
Yeah, most likely it's a transmission electron microscope. Yeah, they're really, it's something that small. It couldn't be a light microscope, and it wouldn't be a scanning scope looking at the surface. And you're right, it probably is a virus. You're viewing a structure that's less than 0.2 micrometers. Does not show any type of internal structure or organelles. What type of microscope are we using here? Probably a transmission. And what's the entity? What are we looking at? Something this small. Probably a virus. And then this is the end of this slide set. I'm saving time for going into the universe. This is something that is relevant to cystic fibrosis, which you're going to see a lot more of soon. But I show this just to emphasize different types of cells within an organ. So the organ here is the pancreas. Pancreas has two main types of cells called acinar cells and islet cells. The acinar cells produce enzymes that help you digest your food. The islet cells produce hormones which help you transfer the energy molecules from digestion into your blood and then into your various tissues. So the pancreas is an essential organ, and without it, you have to go to extraordinary measures to keep a body alive. All right, that's the end of that slide set. I'm going to stop sharing.
I have a question about viruses.
Yeah,
So on one of the slides, it was showing the various viruses. I noticed that the sizes ranged. It was a very big range and difference from one virus to another. Does the size of a virus facilitate how easily it can infect a host cell?
no, they all have really good capacity to get inside a cell. What's different is how they replicate themselves once they get inside a cell, and that's determined by whether their genome is DNA or RNA.
Oh, okay.
So they have very different strategies for that. And when I talk in more detail about coronavirus, I'll illustrate that point. Anything else? Do you see the slide, the universe?
Yeah.
Yes.
Okay. Is there life elsewhere beyond Earth? We are talking about biology, and so we should be interested in other life forms, other life locations beside our own planet. And what I'd like to consider in going through this is what's the likelihood that life does exist elsewhere in the universe? And what's the likelihood that we will ever contact it or it contact us? And so when we think about that, we're going to think about distances. Just to remind you that very small things that we can't see with our eyes, we use microscopes. And some things we can see with our eyes, and then beyond that, very large things that are far, far away, we have to use telescopes. of various types. I showed you this before, so I won't dwell on it. But I want to emphasize distance. Distances are measured in light years. So this is a review. The speed of light is 186,000 miles per second. The number of seconds in a year is 31,536,000. So the distance that light travels in 1 year is to multiply one of those numbers by the other. And that gives you 5.86 times 10 to the 12th. In other words, about 6 trillion miles in 1 light year. And now we're going to talk about, when we get out into the universe, many hundreds, thousands, millions, or even billions of light years. And consider that one of them is 6 trillion miles, and we have multiples of that, just an inconceivably large number. So how do we look at things far, far from Earth in the universe? With space telescopes. So instead of a telescope that's on the Earth, which is encumbered by all the problems with the Earth's atmosphere, a space telescope escapes from Earth, goes out into space and takes pictures. And we've had 2 main ones, the Kepler scope, and we'll talk about Kepler images a little later, and the Hubble Extreme Deep Field Space Telescope. So this is our galaxy. Our galaxy is the Milky Way, and it's massive with respect to our solar system. So here is someone on Earth looking at the Milky Way. And remember, it's a spiral galaxy, and planet Earth is way out here on the edge of it somewhere. So even though it's huge, it's a huge collection of stars, it's tiny with respect to the rest of the universe. And most of them we will never be able to encounter or examine because they're permanently beyond our reach. So even if we traveled at the speed of light, we'd never get to catch up with these galaxies. And I'll tell you why. So this image is a region of the sky that is 1/32 billionth of the total. So it's a tiny, tiny, tiny slice of the sky. Within this little slice, you can count 5,500 galaxies, which is about 10% of the total in the entire universe. The other 90% we can't even observe. So our visible universe contains about 2 trillion galaxies. So 2 trillion is 2 times 10 to the 12th galaxies. If you multiply that by 6 billion stars in a galaxy, that's how many stars are in our galaxy. You get 6 times 10 to the 20th planets in the universe if there is 1 planet per star. And there's probably a lot more than that. So the point I'm making here is that there are many, many planets in the universe, many, many stars, many, many planets. Our Earth is just one of those. So if we're thinking about the possibility of life existing elsewhere, there are lots of opportunities because there are so many planets. But a big problem is this. So these are hypotheses for how the universe began and how it's going to end. So it started with the Big Bang, where there was some kind of an event which released all the matter that we know is in the universe. And you'll notice that the first three of these diagrams refer to a decelerating universe. So in the simplest case, you have all this matter released from the Big Bang. It flies apart because of the force of the explosion. But then because of gravity, over time, all the objects in the universe will have this gravitational attraction for each other, and they'll eventually all collapse together, and we'll go back to the possibility of another Big Bang. Well, I find this intellectually quite satisfying, but this is not what happens. And these other two here are also not what happens. What is happening is that we live in an accelerating universe. So this means that after the Big Bang, things are flying apart, and you'd expect gravity to eventually draw them back together, but it doesn't. Why doesn't it? Well, there's no good explanation for it, except there's this mysterious force called dark energy that's forcing the objects in the universe to defy gravity and actually move away from each other instead of toward each other. So that means that the universe is expanding at an extremely fast rate, and everything in the universe is getting farther and farther away from everything else in the universe. So much of the universe, and there's a huge amount of it that is reachable by us, but most of it isn't. And this is just something to boggle your mind further. So our visible universe contains about 2 trillion galaxies. Most of them are unreachable. And it's because as the universe expands because of dark energy, the space between all objects increases over time. So that beyond distances of about 14.5 billion light years, as I said, we just cannot imagine distances this great, 14.5 billion light years. Space's expansion pushes galaxies away faster than light can travel. So this is illustrated here on the right. The yellow circle shows the observable universe, which is about 46 billion light years across. But because the universe is flying apart, we could only ever possibly access what's inside the purple circle, which is about 18 billion light years across, the purple arrow. So the point that I'm trying to make here is that the universe that we cannot access is huge. Might as well forget about it because we can't access it. The part we can is also huge. And that's where we would expect to find life if it exists. So among these multiple billions, actually trillions of stars, most stars are likely to have planets. So what is the probability of life on them? What conditions are required for life? People who study this, exobiologists, assume that in order for life, at least life of the type we know, you need liquid water, nitrogen and carbon dioxide in an atmosphere, and a modest gravitational field. So first, let's consider water and consider temperature. This is a thermometer going from very cold to very hot. And you see that very cold planets in our solar system would be Neptune and Jupiter. Very hot planets would be Mercury and Venus, both between us and the sun. And Earth here is just right. The water on Earth can be freezing or colder, and it can be boiling or anything in between. And all these other objects that are named here are planets that were discovered by the Kepler telescope. And you can see that almost all of them are out of the range of what would be possible for liquid water. Now, Kepler did find a planet that could conceivably have life on it because it simulates what we find on Earth. So here are two planetary systems, the Kepler-186 system and then our own solar system. Our Earth is 3 orbits out away from its star. Kepler-186 is the 5th planet away from its star. The stars are about the same size. And so the temperatures, because of the distance, the temperatures are about right on both Earth and Kepler-186. And the size is about right. And size is what determines the gravitational force. So Kepler-186 is likely to have liquid water. We don't know, but it's also likely to have an atmosphere. It may or may not have a liquid core to produce a magnetic field, which is important for life. And its mass is about right. If the mass of a planet is too big, the gravitational force is so large that it would crush anything alive. If it's too small, it's incapable of holding the atmosphere. And we already said that an atmosphere is necessary. So these are called the Goldilocks conditions for life. Not too hot or too big, not too cold or too small, but rather just right. So if I ever ask you about Goldilocks conditions for life, this is what I'm talking about. But because there are these Goldilocks conditions, it greatly reduces the number of prospective planets. But that doesn't really matter because there are so many billions of planets to choose from. So the big problem in finding life elsewhere is distance. Just to give you some comparisons, now this is going to be in terms of light years. So the distance from the Earth to the moon, our own moon, is 1.5 seconds. To the sun, 8 minutes. That's how long it takes for light to get from our sun to us. To Mars, 12 minutes. To our nearest star, 4.3 light years. Not light minutes, light years. So our nearest star is unimaginably far away. From Earth to the other side of our galaxy, the Milky Way, is 52,000 light years. So impossible. Now Kepler-186f, this planet that could conceivably, it's similar to Earth, is 500 light years from Earth. That is 2 × 10⁻¹5 miles. It is in the Milky Way galaxy, but the time to get there from Earth would be 500 years at the speed of light. Now we can't go at the speed of light. We can go at the speed of a rocket, which the fastest rocket we have available now, it would take 242,000 years to get to Kepler-186f. So these are impossibilities at the present time. Point I'm making is distance in terms of our search for life elsewhere. Distance is a huge impediment to finding life. What kinds of life have existed on our planet for most of its existence? I've got some pictures of them here. We won't go into them in detail because I'm running out of time. But we can assume that if life forms on other planets like it did here, similar kinds of life forms would be present. And that brings us to another problem. Complex life forms evolve slowly. So here's a time scale. Our planet is about 4.5 billion years old. About 1 billion years after it formed, we had some of the first life forms, single cell life forms. About 1 billion years later, multicellular life forms appeared, and then they got more complex. And then fairly recently in the life of our planet, we've had more complex organisms. And finally, about 7 million years ago, we got ape-like organisms. Humans only appeared about 4/10 of a million years ago. In other words, human type life has existed on this planet for a very tiny amount of its existence, only 0.4 million years, as opposed to the billions of years that life was here, but it was unintelligent. So this means that if there were aliens looking for life on this planet, and they tried to contact life on this planet by sending signals, for example, radio signals, they had no response for over 3 billion years. So any alien that during that 3 billion year time period that tried to contact us, they'd say, this is a dead planet, there's no life on it, because the life had no way to respond. So the only way they could find it would be to actually land on our planet, scoop up some samples and examine them. Similarly, if we're looking for life in other planets, and if life develops in the same scale that it did here, we can't find it because it has no way of responding to us, unless just by chance we happen to encounter intelligent life. But the likelihood of that is really small. So the likelihood of life is much higher than that for intelligent life on any given planet. So the conclusions, I have to go through this quickly because class period is over. So the question we ask at the outset, is there life elsewhere beyond Earth? And the probability of that is very high. And the reason for that is sheer numbers. The number of Goldilocks planets is probably astronomically high because there are so many trillions of planets in the universe. Alternatively, or in contrast, the likelihood of detection by humans is very low for two reasons. One is the distances between planets are simply too great. And secondly, is the point I just made, that if life exists at this point in time, somewhere else, it's very likely to be primitive, unintelligent, so that if we send a signal, it can't reply. So we'd actually have to go there to find the life, and that's confounded by the distance. Distances are simply too great. So will we ever detect extraterrestrial life before we ourselves become extinct? Good question. Don't know. We don't know what new technologies might bring. But one thing we do know is that life on this planet is headed for extinction eventually. Even if we don't destroy ourselves by ourselves, our solar system will evolve and eventually become nothing. So if our species is to continue, at some point, we have to escape from this planet and find somewhere else to go, or build space stations that are self-sustaining. Okay, I've gone a little bit over time. Any questions about anything? I know that I've given you a lot of information and speculation, but it's mostly for you to think about.
I have a question to circle back to the microscopes you showed us.
Uh-huh.
Would it be necessary or sensible for use of one, like 2 or more of the microscopes you showed us to be used in one experiment? Like you showed us 4 of the same cells. Would it be necessary to use more than one microscope?
It may or may not be necessary, but certainly in most experiments that use microscopes, they use more than one type. And there are types of microscope that I haven't even discussed. And so you might start out looking at with a microscope that doesn't magnify very much. And then as you want to find out more and more detail, you switch to a microscope with greater magnification. So answer to your question, and it's a good question, multiple types of microscope are used in most experiments.
Got it. Thank you.
Okay, anything else? Okay, well, that's it for today. You have a couple of assignments waiting for you. And I will send you the recording of this session within about an hour. And don't,
Thank you, Professor.
okay. See you on Wednesday.
Thank
Thank you.
you. Bye.
Okay, bye.